orthobenzoquinone monoketals. So the highly reactive nature of this orthobenzoquinone monoketals are because of that. So we can say two different reactions. One is cyclation, other one is polymerization. So therefore, the polymerization reaction is very slow when compared to the cyclation reaction. So therefore, we have not seen any polymerized products.
Thank you.
So also, we have not used any like distilled styrene. Simply, we have taken the styrene as received. Styrene as received will be having stabilizers like stabilizers for the to prevent the styrene polymerization.
Thank you, sir.
Thank you, Padmavati, for questionnaire session. So now I request Ananta Madam to release the appreciation certificate of Professor Ramakrishna. So on behalf of Department of Chemistry, Osmania University, Hyderabad, this is the appreciation certificate presented to Professor Ramakrishna Peddanti, IIT Roorkee, in the international webinar.
Thank you, madam. Thank you so much.
Thank you, sir. Now I invite Professor M. Vijayalata Madam to say formal vote of thanks, to give the formal vote of thanks. Professor Vijayalata Madam.
On behalf of Department of Chemistry and the organizing members of this international webinar, it gives me immense pleasure to thank Professor Ramakrishna Peddanti, IIT Roorkee, for his excellent talk on recent advances in the synthesis of carbo and heterocyclic chemistry. He being my senior in HCU, I know him very well, but according to this talk, he has talked about this catalytic asymmetric synthesis and their biological relevant molecules. Apart from that, he also talked about the green synthesis, where he talked about the solvent-free, high-efficient Diels-Alder reactions. And by simple grinding mechanism, you can get the benzoxazenones by reacting orthoaminophenol with acetylene dicarboxylates. And these obtained benzoxazenones can be reacted with nitro-derivatives, parabenzoquinones, 1,4-naphthaquinones. And also he talked about the 1-pot domino oxidative cyclization acetylation Diels-Alder's reactions. Finally, to conclude, he has talked about many reactions which are very, very relevant for our organic students, and it will be an insight to work on Diels-Alder's reaction to give excellent products to be obtained and which can be published in good journals. Thank you very much, sir, for your excellent talk.
Thank you, Vijayalata. Thank you very much, sir. Ramakrishna, sir. Thank you very much for being with us to give the more informative lecture, for sharing your research to us. Thank you, sir.
Thank you.
So coming to the last session of today's webinar by an eminent speaker, Dr. Padmaja Juvadi, Director, Quality, United States of America. Is she there? Now a floral welcome to Dr. Padmaja Juvadi. And she is an alumnus of Chemistry Department, Osmania University. And I'm glad to say that she was my senior and my good old friend. Now I request B. Sirisha to kindly introduce Dr. Padmaja Juvadi.
Good evening, everyone. Am I audible?
Yes.
Yeah. Very good morning, Padmaja madam. It gives me immense pleasure to introduce our proud alumni, Dr. Padmaja Juvadi. I'll be sharing the screen. Madam Padmaja has completed her MSc in 1982 from our department and completed her PhD in 1987 in synthetic organic chemistry, guided under the eminent faculty, Professor C.V. Ratnam sir. She has resumed her postdoc at Indian Institute of Science, Bangalore in 1987, and she has joined as the faculty in University College for Women, Koti, and later left the job for the postdoc at Washington University, US School of Medicine with Professor Garland Marshall from 1990 to 91, and later the postdoc at the Rockefeller University with Professor Brice Merrifield, a Nobel laureate from 1991 to 97, and a co-investigator on NIH grant. And to her scientific credit, she has published the peer-reviewed papers in the national and international journals, including the Journal of American Chemical Society. Later, she worked at different biopharmaceutical companies and was instrumental in launching the pharmaceutical and biopharmaceutical products in US, Europe, India, and Japan. She has hosted and supported the regulatory investigation and inspections by Food and Drug Administration, FDA in US, and Drug Control General of India, DCGI in India, and European Medicines Agency, EMA regulatory in US, and Health Canada in the Canada, etc. With this brief introduction, now I invite Madam Padmaja to deliver her lecture.
Thank you,
Thank
Sirisha. We welcome Padmaja Madam to enlighten us with her. Padmaja Madam.
Good morning, good evening, everybody. And thank you very much for your kind words, Professor Bhavani and Professor Sirisha. I'm honored to be here and share some of my experience with you. Since we have heard quite a bit about the chemistry and how the synthesis occurs and how we identify the candidates. Now I'll take you through the journey of taking the lead candidate and going through the non-clinical, clinical trials and the FDA approval. When I say FDA, it's like any regulatory agencies, but just for the sake of convenience, I use the word FDA, which is the US regulatory agency. We'll walk through the journey of the drug approvals. Let me see if I can share my screen. Are you able to see my screen?
No, it is not.
No,
Is it?
Are you able to
screen
see my
share
screen?
has not yet started actually.
Not
Not
yet
yet.
started. Yeah.
Sorry about the technical difficulties. I'm not able to, okay.
At the bottom, share screen button
Yeah,
is
yeah,
there, ma'am.
Yeah,
Yes.
it is getting
I see
started.
my screen now.
Yeah.
So as I mentioned, I'll go or I'll give you an overview of the new drug approvals from a quality perspective. So before I go into the details of the process itself, let us just go over the meeting topics, what I'm going to talk about. So basically, I will start with an overview of the drug development and approval process and talk a little bit about the clinical trials and FDA review processes and challenges and faced during this process and the risk management and the applicable regulations or the guidance, and and then I'll conclude with the inspection findings. So when we talk about the drug development and approval process, what actually is the drug? So drug from an FDA perspective or from a regulatory agency's perspective is defined as any product that is intended to be used in the diagnosis, cure, mitigation, treatment, or prevention of disease. Or it can also affect the structure and function of the body. So the next slide talks about this. It's a busy slide, but it basically walks us through the whole drug development and approval process. As we have already listened to the earlier very interesting talks where we have synthesized the product compounds. And once we have identified the lead, now I'll walk you through the process of approving the drug and putting it into the market. So once we identify the lead, then based on the initial product profile, it will be identified as a candidate to further pursue the non-clinical and the clinical trials. So and then the candidate goes into the non-clinical trials. So non-clinical or preclinical, also called as preclinical trials. So these trials are conducted in animals. The reason why we do these trials is to understand the toxicity of the compound. And we try in different models based on the product and also the profile of the disease. So we try in different models for the non-clinical. And once we get the data from the non-clinical or the animal studies, it is in favor, like the product is not really very toxic, and it seems to be effective in the animal model, then we move on to the first in human trials. But before getting into the first in human trials, we have to do something called an IND. So what happens is, once the non-clinical study is completed, we do a non-clinical report and have a pre-IND consultation with the regulatory agency to discuss the issues that we identified during the non-clinical study, and also to align with our thought process on how we wanted to go ahead with the clinical trials. So then what happens is, it is a must that the IND should be approved by the regulatory agency before FDA, before we get into the first in human. So I'm just trying to make it as simple as possible because there are multiple steps which are involved, even though it is even if when we have to submit our IND to FDA, we still need to have the pre-IND consultation. We have to have identify an IRB. IRB is an institutional review board, which is an independent body. And it's not connected either with the sponsor or with the FDA, but it ensures that the ethics are maintained during the clinical trial. And it plays quite a bit of a role in the whole process, where it reviews the clinical protocol, it reviews the informed consent form, and it ensures that we follow the rules and the regulations given by the FDA. So the other thing is when we have this pre-IND consultation, we discuss the animal data, and we also modify the target group based on the toxicology data. But the bottom line is we are aligned with the FDA's, and our thought process should be kind of blessed or aligned with the FDA's requirements. And then we go into the clinical trials. So the clinical trials, first we go first in human trials. I'll talk a little bit more about the clinical trials in next few slides, but just to give overview of the whole drug development and the approval process, first we do the phase 1 trials, first in human trials, followed by the proof of concept, which is the phase 2 trials, and then to the phase 3 trials. And of course, then we have done with our clinical trials, everything goes well, and we're ready to submit our application to the FDA. So this is called a new drug application, NDA. But prior to filing, we again have a discussion or a meeting with FDA and make sure that our data is acceptable, like a high-level discussion where we give what are the issues we have faced during the clinical trials, and whether the data supports the proposed labeling. And that's one of the major important factors that FDA needs to agree upon. And prior to launch, once the FDA, we submit our NDA, FDA reviews it. And again, I'll go over the review process a little later. But once the FDA reviews and thinks that the data is acceptable and the product is ready for to put it into the market, that's when we are eligible to launch. So, but however, the NDA approval is a must. So once we launch, does not mean that everything is done. We are committed from a sponsor's perspective. We have a commitment to do a post-market surveillance. Though we do the clinical trials like phase 1, 2, and 3, it is still a limited where we are testing our product. And once we put it into the market, it's an obligation from the sponsor to make sure that the product is still safe in a wider population and basically different geographical distribution. So there is a commitment that needs to be done, and it is called the phase 4 study. So then I'll go to the next slide where we talk about what the clinical trials are. It's basically a clinical trials or the lead candidate, which we have identified during the synthesis and put it into the non-clinical study. And then now we are ready to put it into the humans. So why do we do that? Why do we have to do that? It's also to understand the clinical pharmacokinetic and the pharmacodynamic effects of the product, and identify if there is any adverse reactions with the product in human. And also, once we give the product to the people, we study the absorption, distribution, metabolism, and excretion of, and understand more about the product. And the whole clinical trials, the intention of these clinical trials is to ascertain the safety and efficacy of the investigational product. And the next slide gives a little more details on different phases. So when we are talking about the phase 1 trials, as I already mentioned, it is the first human trials of a new drug or a new drug formulation. And these trials are carried out in healthy volunteers. The reason why we do that is to evaluate the safety of the drug, and of course, understand the initial profile of the PK/PD profile of the drug too. So phase 1, sometimes people also do something called a phase 0 study, which is like basically an exploratory study. And it's done in very limited number of patients, probably like 3 to 5 subjects. And that is to understand the profile of the product. Instead of going into the, like if you're not, if you wanted to understand it better before getting into the phase 1, or as you're waiting for the IND approval, some sponsors do that so that you will have more data before even getting into the phase 1 study. And then once everything goes well with the phase 1, we go into the phase 2. The phase 2 is actually done in the patients, in the diseased patients for which we are targeting our product. And the reason why we do that is to evaluate the safety of the product, of course, and also to determine the efficacy of the product in patients. And the phase 2 trials are again designed in a smaller number of people or subjects. And it's often done in a comparative manner. The design is like one of the arms will be the placebo controlled and the other one will be the drug. So I'll go into the details of what placebo is in my next slide. And this, doing the phase 2 and getting the efficacy results will also help us determine what the dose will be for this product in order to be efficacious, but safe, and no or very little adverse reactions. So it's always, we have to weigh in the benefit and the problem with the drug. So the next one is about the phase 3 and phase 4. Phase 3 is more or less similar to phase 2, but in a larger populations, and it's more geographically distributed. So therefore, there are more number of people, and naturally, we will see we may see more number of side effects. But this is for us to understand, again, the safety of the product and the efficacy of the product. And these trials are preferably randomized and double-blinded. This is again to ensure that we are not biased with the results. Again, I'll go into the definitions of what randomized and double-blind is in my next slides. So, and the other requirement or expectations with the phase 3 trial is it should be conducted as close as possible for the real world. Because once the phase 3 trial is done, we are requesting FDA for market authorization. So that means this product gets into the market and people will use everywhere. So that's the reason why it has to be, or it is expected to be, as close as possible. And then comes the phase 4 study. As I mentioned, it's a post-marketing surveillance. And these are performed after the pharmaceutical product is launched in the market. And then during the process, sponsor is obligated to collect the safety information and monitor the adverse events. And the FDA actually maintains a program where the post-marketing surveillance results are available for everybody, basically. And they all monitor it, they trend it, and if they, during the course of the time, if they find that there is an issue with a specific product, then they would recall the product. Like as an example, we all are aware of Zantac. Zantac has been in the market for a long period of time. But during the post-marketing surveillance program, there were issues that were identified, and they have to recall the product. So that's an extreme case. But sometimes what happens is when they find that there are issues with the product, then they would modify the label, and they would put a box on the label saying that this cannot be used for children under 14 years, or this cannot be used for women who are pregnant. So those are the things based on the safety data that has been acquired during the post-marketing surveillance, the modifications happen. And it's not uncommon to have the label modified in the course of the life cycle of the product. So the next slide basically defines the different types of clinical studies which are done in a normal way. So when I talked about the randomized trial, what does it mean by randomized trial? So that means like when the subjects come into the clinical trial, they are randomly assigned with one or two arms of the study. That means this is only to avoid bias again. So when they do the pre-screening, they do not want the sponsor to already think that, oh, okay, these people have XYZ issues, therefore I'm going to give him or her higher dose of the product. The sponsor is not allowed to do that. Therefore, we do a randomized study. And when we talk about the placebo-controlled clinical study, generally there will be like at least two arms, like in the phase 2 and phase 3, there'll be multiple arms, but a minimum of two arms. Whereas one arm is the placebo and the other one is the active group. That means the placebo is exactly the similar as the active with all the formulation and everything, but the active ingredient is not present. So this allows the sponsor to understand if the product is really efficacious or not. Or is it just the formulation that we are giving? And because we are giving something, the patients or the subjects are feeling better. So that helps us determine the efficacy of the product. And then we have something called an open label study. So this means everybody knows what's going on with the clinical study. So the patient is aware what drug they're receiving, and the patient is also aware whether it is a placebo or the active drug, and the investigator is, of course, aware of it. And then we talk about the blinded and the double-blinded studies. So the reason why the blinding is performed is to avoid bias. So when the patients are not told about what they are receiving, whether they are receiving the smaller dose of drug or the higher dose of drug or the placebo, patient is not aware. They just receive something. And the double blind is neither the patient knows nor the investigator knows. And this is very critical to have a double blind study in the phase 3 trials. What happens is during the trial, if say, for example, there is an issue with, there is an adverse event of a subject. So neither the patient knows nor the investigator knows. So what is given to that particular patient? So then there will be a group of people who are unblinded. And those are the ones who will break the code, saying that, okay, this person is being given higher dose of the product. So then the reason why they break the code and give that information is the highest priority in any of these trials is to ensure that the patient is safe. So if there is an adverse event, you break the code, you understand what the drug is, what dose and whether it is a drug or the placebo, and then see if the reaction is associated with the drug. Sometimes even with the placebo, there might be an adverse event. So that means it is not related to the drug, but it is related to some other ailment or a pre-existing condition of the subject. So that will make us understand it. Again, the endpoints and the outcomes of the clinical trial need to be set and defined in advance. So our IND clearly mentions what kind of outcome are you expecting? What is your endpoint? What is your primary endpoint? What is your secondary endpoint? And what is your exploratory endpoints? So when I say primary, secondary, and exploratory, you determine whether, say, for example, if the drug is being used for COPD, so what is the endpoint? Is the subject able to feel better, or is the COPD has decreased? That's what that could be a primary endpoint. And the secondary endpoint could be, is there the quality of the life, did it increase? And then the exploratory endpoint could be, you still collect the blood and do other research-oriented exploratory studies of that particular sample. The reason why you do this is to understand the profile of the product. But in order for us to do the exploratory studies, it is imperative that it is mentioned in the informed consent form, and the subject approves or agrees that we could use the sample for the exploratory purposes. So again, I cannot stress the importance of the informed consent enough, but it is very, very important to ensure that we get an informed consent from the subject. And when you say we get the informed consent, it is also the sponsor's responsibility to make sure that they understand what the informed consent form is saying. So the language has to be very simple, and sometimes there is a need for interpretation. And sometimes there is also the language barriers. So these are all the things which sponsor needs to take care of it. And the informed consent form should be, must be approved by the IRB, which is again the Institutional Review Board. And its role is to protect the subject's safety and also ensure that the sponsor ethics are followed. So as an example I gave here as an overall survival rate of the toxicity. Sorry about that. Sorry about that. So I kept on saying safety, efficacy. So what exactly does safety mean? Safety is the highest tolerable dose that could be used to achieve the desired clinical benefit. So say, for example, let's take the simplest example as the Tylenol. Tylenol comes up in like different dose formage. So the 325 is the minimum, the smallest dose for adults. And people can use 1 tablet or 2 or 4. So they do say that on the bottle. You could use 2 tablets every 4 hours. So that means we have already tried those requirements in the clinical trials to say that. So that's the reason why I mentioned that the labeling plays a major role in the final approval by FDA. And coming to the efficacy, what is efficacy? So is to whether this product is has any effect on the disease, or is it just I'm using it, there is no effect. So that's what is determined. So basically it is whether a drug has a positive clinical benefit over placebo or any other intervention. So these are all I tried to cover most of the types of clinical studies, and so these are all just to give you all an overview of what goes on in the clinical studies and how there are different ways to do the trials and how important it is to ensure that the patient is safe in the whole process. So this kind of consolidates all the phase 3 trials with the non-clinical and also the FDA review. So as we have discussed in different ways from the beginning, so the non-clinical studies or testing is done on animals for toxicity. And it is, once the toxicity data is achieved, it is required that the investigational new drug application is submitted to FDA, and IND approval is a must to initiate the clinical trial. So, and then we go on to the phase 1. So here we check the drug for safety, and these are all the healthy volunteers. And the number of, number of the healthy volunteers could differ, 20 to 60 subjects. And within the phase 1 also, the sponsor plans to do phase 1A and 1B. Which one is like the 1A is sponsor gives the product only as a single dose for a particular number of patients, for a particular number of healthy volunteers. And then for the 1B, they give as in multiple dose. And also they do then ascending dose escalation. So, and here we could find some unexpected side effects during the phase 1 trials. And once the phase 1 is completed, sponsor submits the phase 1 report to FDA, and followed by sponsor and FDA could have a meeting, or it is recommended to have that meeting so that you can discuss the phase 1 report and also discuss the phase 2 study plan. The reason why you do that is from the very beginning, it's a collaborative effort. From the very beginning, you are aligned with the FDA's thinking. Therefore, there are not like total surprises at the end of the trials and when you submit the NDA. And with the phase 2 trial, then we start our phase 2 trials where we check for the drug efficacy. And this is a smaller number of patients, like 100 to 300. And most failures are identified in phase 2 trials. The reason being, the drug is not as effective as we thought it would be. The phase 1 is the safety because we already did the non-toxicology studies in animals. And so we have some kind of information as far as the safety is concerned. So therefore, when we come to the efficacious trials, which is the phase 2, there is most failures occur at this. And again, at the end of the phase 2, we submit in a phase 2 report to FDA and discuss the plan for the phase 3 study plan. So the phase 3, phase 3 is an extension of phase 2 study where we confirm findings in more number of patients. And this can range from 1,000 to 3,000 patients and geographically distributed, not just in US, but in EU, based on where you intend to launch the product. So it is geographically extended. And since we did that, it could detect more side effects, or it could move on. And that would be the best scenario. When everything works well in phase 3, great. Now we are ready to submit our new drug application to the FDA. So therefore, we submit the NDA or the BLA based on whether it's a small molecule or the protein or the biosimilar, we submit that. And then it's on the FDA's court. So FDA has all this data, though they have been involved from the very beginning, but not thoroughly. FDA does a thorough review of everything we have submitted. In the sense like they audit the data, they make sure that it is conformed with the application and there are no glitches, there is no errors, and they confirms the safety and efficacy of the product. And also they decide if they have to conduct a PAI, that's a pre-approval inspection. So once they're satisfied with our application, once they have conducted the pre-approval inspection of the manufacturing site and or clinical sites and the sponsors too, then they finally give us the approval. So once it is an approved drug for marketing, hey, so that's the best time of anybody's life, I would say, to receive that letter of approval with all the effort we made so far. So now we're talking about the FDA drug approval. So let's see what FDA, what happens. So the FDA follows a specific path and it has established, and the agency ensures that the drugs are safe and effective when approved. So in a way, we do feel like FDA takes a lot of time to review this one. Why don't they do it faster? But we have to understand they have a bigger responsibility because the health and safety of people is the prime responsibility of the FDA. They do want it to make the medicines approved, but once they approve it, we are free to launch the product, and the product is available for people to use it as they need it. So, so there are two critical approvals as far as FDA is concerned. Early stage approvals, which is an investigational new drug application, IND approves. And at this time, agency reviews the information coming from the discovery through the non-clinical studies, so the toxicology studies, and in order to initiate the clinical trials. And the late stage approvals is a new drug application where they review just to put you into the perspective, when we're talking about new drug application, it's at least like 1,000 pages, which includes the animal data, human data, manufacturing data. So the manufacturing data means like manufacturing of the drug product, drug substance, and also the testing, specifications, labeling, and everything. So it plays a major role in the whole approval process. And once the NDA is approved, we are free to launch the product in the market. So again, this slide kind of gives what happens in the FDA arena. As I mentioned, it takes a lot of time for the FDA to review and approve. Initial NDA submission to final approval varies approximately 10 months. But FDA's goal under PDUFA priority review is 6 months. So let me walk you through what happens in between FDA and sponsor during this process of 10 months or 6 months, or whatever the case may be. So we have a pre-NDA review meeting with FDA, where FDA and sponsor meets before submitting it on the NDA and aligns with the data what we have acquired so far. And sometimes in that meeting, FDA says, oh, maybe you should do some extra study, confirm this particular bioavailability or whatever, based on the data we have submitted at a high level. And so that it is really critical to have that pre-NDA meeting. And once we align, the sponsor and the FDA aligns with the NDA content, sponsor submits the NDA. So NDA basically includes animal, human data, and information on the drug manufacturing process and testing, release, and all the data. And it also includes the information on the facilities used, CMOs we use, and how are they qualified, what kind of quality management program do you have with the sponsor, and whether the audits are done. So all this information goes into NDA. So once the sponsor submits the NDA to FDA, FDA has 60 days to file NDA. So that means that's an initial acceptance. So what they check in that 60 days is technically whether it is acceptable. Did you have all the data which is supposed to be in there? Not the thorough review. It is only the preliminary review to accept the application. So the reason why I repeat that is sometimes people get misled that initial application means FDA approval. No, that's not the case. It only means that FDA had accepted to review your application in order to approve the product. So then you get a day 74 letter. It's called a day 74 letter. That's when the sponsor receives a letter saying whether FDA had filed the NDA to review or refused to file. So say we think that it is, they have accepted our application. So then we move on to the next step. So the expectation is in 120 days, like 4 months, sponsor is expected to provide with a safety update. Remember, we are saying that the long term, the phase 3 trial is over, but we are still continuing to see how the subjects are doing. Is there a safety issue that needs to be shared with the FDA and all that? So 120-day sponsor submits the safety update. This is also important for FDA because based on this safety update, FDA will ensure whether it matches with the labeling, labeling is accurate or not, or is there a need for modification. And in the month 5, there will be a mid-cycle meeting. This mid-cycle meeting is an internal FDA meeting. And after the meeting, the project manager communicates with the sponsor. So what the communication includes is mainly the 3 aspects: risk evaluation and mitigation. Is there strategies? Is there any risk associated with your application? And how are we going to mitigate the risk, and what the plans are? And what kind of post-marketing requirements they are thinking that is required, and do the sponsor agree with those ones? And they also may or may not mention about the pre-approval inspections, but they can give an idea that they might be doing a pre-approval inspection. And then they continue reviewing. So this communication happens within the project with the project manager and the sponsor. And actually FDA recommends, and with any kind of questions the sponsor may have, to discuss with the review panel so that it's clear. And they have somebody raised hand.
At
Am I
the
supposed?
end, you can answer.
Huh?
At the end of the lecture, you can give answer.
Okay. All right. Sounds good. And then there will be a late cycle meeting, which is like close to the completion of the NDA review. So during the late cycle meeting also, then they will further discuss if there is any risks associated with your product, and if they need more explanation or they ask you for more data and all those things. So once this happens, so FDA then goes to the official action. So what is this official action? So in the late cycle meeting also, they could mention that they are intending to pre-approval inspection. It is not required, but they generally mention their thoughts, or they share their thoughts for a need for a pre-approval inspection, so that the sponsor is prepared, and the sponsor prepares the clinical sites, and also the sponsor communicates with the CMOs if somebody else is manufacturing the product for them. And then comes the official action, the big day. And so you get in a letter from FDA. It could be 3 different categories. One of them is an approved letter where it says that everything is fine. We agreed to this risk management strategies, and we agreed to these post-marketing commitments, but you can go ahead and launch. But then the second letter, second type of letter is an approvable letter, which talks about your data seems to be good, the product is safe and efficacious, but we need to confirm some other things. And therefore you need to do XYZ things in order for us to consider it as an approved product. So that would be our approvable letter. And then the last and the least you would want it is a rejection letter. So, but it does happen sometimes. And okay, so what is the overall probability of this success rate for the drug, starting from like going through the whole process of drug development, from the phase 1, like non-clinical through NDA? Actually, as I mentioned earlier, the highest failure rate occurs in phase 2 testing. Almost 60% failures occur at phase 2. The reason being, this is the first time we are testing if the drug is efficacious or not. And unfortunately, we do find quite a few times that the drug is not as effective as it is supposed to be. And so it's interesting. I mean, every time I see this, it surprises me. Overall probability of a drug passing all steps is only 11%. So there's a lot of work goes into the whole process, but there is only some chance, like, so basically 1 in 10 will be approved and the others don't. And the time it takes for this whole process is like 10 to 12 years or 10 to 15 years, approximately 12 years. Again, it depends on the disease, what we are targeting. But again, the whole process takes a long time. But we just have to remember, once this process is done, the product is in the market. And that means a lot for the sponsor, for the subjects, for regulators. So there's a lot of coordination that needs to happen in order to bring the product into the market. So what are the challenges during these clinical trials? So the challenges may be systematic, and that can render the data to be unreliable, or the errors can be specific. The study design, we have not designed the protocols properly. Therefore, what happens is the protocol does not clearly indicate everything. Therefore, there will be quite a few errors made at the clinical sites. And the investigator is not clear, the monitors are not clear on what exactly we meant. So therefore, there will be a lot of deviations to the protocol that will come up. So once we identify that the protocol, there is an issue with the protocol, the best thing is to amend the protocol. So I'll give you an example. Like if there are some issues when executing the protocol in 1 or 2 sites, but all the other sites, all the 50 sites are doing or understanding it well. So that means it is the site issue. But if there are issues, similar issues with like more than 50% or more than 20% of the sites, that means the protocol is not really clearly explaining what needs to be done. So the design fault is there. Therefore, you go back and amend the protocol. And once you amend the protocol, it again has to go through the process of approvals. And if it's a simple amendment, it still needs to be approved by IRB, and it needs to be informed to FDA, or it needs to, based on the changes you make, it either needs to be informed to FDA or again approved by FDA. So as you see, then again, it takes a longer period of time. Therefore, it is really important that the protocol design is done accurately or appropriately, and the clarity is in there. And then the other errors come due to the study conduct and monitoring. the clinical investigator sometimes do not provide enough oversight. They rely on the monitors or they rely on the co-investigators. So that might lead to compromise the patient's safety, and also it can result in the unreliable data. So, and then the sponsor actually sends the monitors to monitor the clinical site. And sometimes the monitors do not do a good job, or do not escalate the issues in the right time. Therefore, the issues keep coming up, or issues keep adding up. So, and then the errors can include with the data storage and management. So there are no internal processes or inadequate internal processes where the quality has an oversight. So then that results in the issues with the data reliability and use the use of the data. And then there could be issues with the data analysis. So the statistical analysis could be faulty, and the statistical analysis plan does not really reflect what the protocol is saying. So therefore, there will be the issue with the data analysis. So all these types of errors needs to be addressed with a good quality system. In spite of we talking about the issues, understanding the issues, putting the quality measures, there will still be some issues. But as long as the data is reliable, and the most important thing, the first important thing is as long as the patient's safety is not compromised and we get the reliable data, it is known that there will be some issues. But when I'm saying some issues, like minor ones, it cannot be some systemic, or it cannot be a study design. And so then we need to make the corrections. So how do we do this? So first of all, with the clinical trials, it generates knowledge on the safety and efficacy of the drug. But the scope of the knowledge differs. In academia, it generates the knowledge. There's a lot of knowledge that comes from academia, and the regulators basically assess the knowledge. And the industry, pharmaceutical industry or the biopharmaceutical industry, use this knowledge in developing, testing, and marketing. So it's basically from the lead is all coming from the academia, mostly coming from the academia, and also some industries do the research, but it is the generating the knowledge. And the industry is basically using it to develop and market the product. So all these issues, what I just spoke about, the probable quality related issues, can be addressed having a risk management plan in place. So that's kind of a way to go, where in the risk management plan, this is kind of a live document where we say that, okay, these are the risks. In the beginning, we do say that these are the risks associated with the whole program, and where we can think that there can be a problem. But as we are doing the trials, we identify different kinds of risks. So we need to list those risks, modify the risk management plan, and have the mitigation strategies, risk mitigation strategies, to ensure that we address those risks. Again, the bottom line is to ensure the safety of the subjects. And as I said, I cannot really insist on how important it is to ensure that the safety of the subjects is taken care of. So again, what values do we stand for? And how do we bridge the gaps? We have all these issues. And how do we do this? Well, what are we going to address? By building quality into clinical trials. So how do we do this? Quality by design for clinical trials. So earlier, in olden days, I would say, the approach was quality was coming at the end of it, end of the study, or are mostly coming at the end of the study, where they're reviewing the data and saying that this is good, this is an issue and all that. But the approach have changed quite a bit. And the expectation is to build the quality from the beginning, from starting with the protocol development itself and going through every aspect of the trial. So during the monitoring, if there are any issues, quality comes into picture and addresses those issues. And if the issues are not resolved with the monitoring process, then quality does a for-cause audit of the site to understand if there are any other issues which are deeply embedded and that might compromise the data at the end of the trial. So, and then focus on the key risks to ensure that the trial integrity is not lost and the data quality is maintained, and of course, the subject safety. And the reason why the regulators are encouraging or expecting the quality to be from, like, through the clinical trials itself, quality by design approach is so that you can detect, the sponsor can detect the problems and correct them in real time, rather than go through the whole trial, spend all that money in each phase of the clinical trial, and then realize, oh my God, we should have done that in the beginning. So that's the reason why quality plays a major role in detecting and correcting the problems and escalating the problems and ensuring that they are addressed appropriately. And of course, with all these things, whatever we do, we need to adhere to the regulations, follow the requirements, GCPs and the GMP requirement. GMPs are mostly for the manufacturing, IMP manufacturing, distribution, and storage, and the GCPs are more for the clinical trials, and the GLPs are for the non-clinical studies. So I'll go ahead and touch base on the regulations now. So good clinical practices. What is it? It's a standard for the design, conduct, performance, monitoring, auditing, recording, and analysis, and also reporting of the trials. And it is required, GCP requires that we provide assurance that the data and the reported results are credible and accurate, and the rights, integrity, and confidentiality of trial subjects are protected. So that's ethics. So basically, to be in simple words, quality data plus ethics is GCPs. So what we need to think from an individual perspective is, how do we generate quality data? And how do we ensure that we meet the ethics requirement? And then that means we are following GCPs. And so I just put a slide where it gives you a flavor of the history of GCPs. As you all are aware, I don't have to go into the details of it, but the theme or the requirement for the GCPs or the GMPs is the rights, well-being, and confidentiality of trial subjects are protected. Trial data is credible, and it's scientifically sound studies are conducted. And also, do not repeat mistakes. So from the timelines, as you can see, it started from 1906, and where we earlier until 1938, it was only like the safety of the product was important. But then the sponsors were claiming whatever efficacies they wanted to say because it was not going through the FDA's. And now FDA has become more stringent where the efficacy is important, and the sponsor is responsible to ensure that the trial is conducted on an ethical standard. And then the ICH guidelines came into picture, which has become law in 1997 in some of the countries, but it's mostly of the guidance. And I'll go into the details of it, but in 2016, the GCP E6, which is specifically for the clinical trials, has been amended, and an addendum was added to the E6, which is called an R2, which actually talks more about, excuse me, more about the quality involvement. So this is like the FDA regulations for clinical trials. Again, I only listed the different regulations that needs to be followed. And I'm not getting into the details of what each regulations are required due to lack of time, because that's another webinar, I would say. To summarize the FDA regulations, all the enforceable US laws are housed in Title 21, and which requires the minimum requirements for the conduct of the clinical research and the manufacturing and distribution of the IMPs, investigational medicinal products. And these are all the parts 50, 56, 54, and 312, which basically is directly related to the clinical trials. And as I mentioned, 56 describes the requirements and the responsibilities and the purpose of the institutional review boards. And part 11, you all are aware, the electronic records and electronic signatures, which has become more prevalent. And part 820 is the quality systems and risk management. And 210 and 211 are more for the CGMPs, where need to follow in order to manufacture the product under the GMPs and do the testing and also distribution. So I'll go over the GMP regulations. I don't know what's happening with this, but hold on, please. Okay. Hold on again, sorry. You did, okay. So the 5 major concepts of GMPs is safety, identity, strength, and purity, and quality of the product. Again, as I mentioned, it's 21 CFR 210 and 211 that that talks about the GMP requirements. And again, as I mentioned, the ICH is the guidance which is used internationally by almost all the countries. I'll talk about the members of the ICH, but right now, so the ICH has developed, say for example, more than 60 guidances. And the 4 main topics they talked about is the quality, safety, efficacy, and multidisciplinary. So when I'm talking about the multidisciplinary, basically those are the topics that do not fit uniquely in one of the above ones. And as we already talked quite a bit about the quality, safety, and efficacy, I'm not going to get into the details of that, but just want to point it out what the efficacy E6 guidance for good clinical practices is the most important one. And to be honest with you, mostly in the internal documents and internal SOPs, E6 is the one which is referred the most. And the guidance are valuable support to develop internal procedures and also to develop the clinical protocols, CSRs, statistical analysis plans, and follow them so that it's easier for response from a sponsor perspective, excuse me. So then let's just touch base a little bit on the E6 guidance. So the definition is, again, I'm repeating this, but I wanted you to take home that it is an international ethical and scientific quality standard, which is designed for designing, conducting, recording, and reporting trials, which involves the human subjects. And the purpose is global standardization. So the FDA regulations, again, in India, we have the DCGI regulations. It's only applicable to the local regulations. Though we follow the ICH guidelines, we are still obliged to follow the local regulations if we decide to launch the product in the market. So the purpose is the global standardization and then the protection of subjects and and the data integrity and the quality. So why did we revise in 2016? The reason, generally the revisions happened because there was some issues that was identified, or there was a request from sponsors and from the industry personnel asking for clarity. And in this case, from the R2 addendum was added because the European Medicines Agency, EMA, had submitted a report in 2014 summarizing 398 GCP inspections, which were conducted in the 2000 to 2012. And the critical and major findings were mostly in relation to monitoring, data management, clinical study reports, and source documents. So, and therefore, they have ICH has revised to address the GCP inspection findings and these were addressed by introducing the quality by design approach and quality risk management approach. And the focus is also on the technological tools to ensure that the robust conduct, oversight, and reporting. When I say technological tools, is earlier when the subject was going through or participating in the clinical trials, there were like patient diaries where they used to write down the medication taken and then bring it back to the sponsor. But now it is the e-diaries are in there. So that means you need to make sure that it is a validated system. The patient is not entering something and we are seeing something else. So, and also how to protect the confidentiality information. Though we go through the whole trial, the patient's name or the age or, like the personal information is all blocked. So that's how the focus is on the new technological tools. So this is I was talking about the critical findings. This is a summary of the critical findings. I'm not getting into the details of that, but it's mostly the monitoring, data management, CSR related, protocol compliance. So the protocol, especially it is the selection criteria and the source documentation. So when I keep saying about the source documentation, it is the actual data. It is not the report or not the data analysis, not the report from the data analysis. The report from data analysis gives us an overview of what has happened. But FDA goes through the source documentation and makes sure that the data is what we are reporting. So that's one of the reasons, they audit the data. They look into the source documentation and they also see, as an example, let me go from a chemistry perspective. We do submit HPLC data, or we say that, oh, by HPLC or by mass spec, this is the molecular weight of the product. But now they wanted to see actual the data that is coming out of the instrument, the real data. And they look at that and then confirm what you said is true. So that's where we're talking about the source document. And from a clinical perspective, the clinical diaries, the one which patient writes on it is the one which is the source documentation. And when the investigator looks at when an ECG is done in the laboratory during the investigation, that is the source documentation. So I think I kind of touch base with both the GCP and the GMPs, source documentation. And as I mentioned, these are all the ICH members. These are the founding members, which started with the European Commission and the United States and Japan. And Health Canada and Swissmedica, or these are all the regulatory members, and ANVISA, and just have to note that the pharmaceutical representatives also participate in the discussion And they also make suggestions on how to revise this, what kind of explanation needs to be given, and all that part of it. So these are some of them which I listed here, which involve in the decision-making or policy-making process during the revision of the ICH guidance. So now let's see that it's a question time. Why GCPs? According to the principles of the ICH GCP or the FDA GCP, what is the most important consideration when conducting a clinical trial? Is it data accuracy? Is it the protection of the trial subjects? Or is it retention of records, reproducibility of the trial? So as you think about it, I'll give you the answer. It's the protection of the trial subjects. So as I mentioned several times, that's the most important. The right, safety, and well-being of the trial subjects are the most important consideration and should prevail over interests of science and society. So the expectation is, first, do not do any harm, and then give me a medication for the treatment of the disease. So the reference is, again, ICH guidance and all of the FDA guidances, which regulations, which I have already pointed out. So this is, again, the GCP question training. There are so many different issues that can arise. So I just wanted to walk you through like the issues that can come up during the clinical trials, but can be addressed with a proper quality management and the risk management of the issues. So this is just to give you a flavor of few of them. I could not list all of them, but these are the few of them which can come up in the whole process. And it could start with the financial disclosures. So like, there should not be any interest for the investigator to conduct the trial. So that means there is a bias. So they have to give us a financial disclosure saying that we are not getting anything from the sponsor. Sponsor has to account for every penny they use during the trial if they are providing any incentive to the clinical sites. So those are the things, just to point it out. Again, like, do you have an audit? Do you have to audit every vendor? Is a question that comes up. No, you don't have to audit every vendor, but you have to have a risk assessment of each vendor. So why am I not, they say, for example, if I'm doing clinical trials in 100 different sites, I'm not going to audit all the 100 sites, but I'm going to have a plan on why I'm auditing these 2 sites and why am I not auditing those 80 sites. So, or give an explanation and support for your decision for not auditing them. So I can go on and on, but I think I'm already running out of time. So let's keep going. So for all the clinical trials, investigator plays a major role. And as you can see, these are all the ones, these are all the regulations related to the investigators' responsibilities. They have to be qualified. Their primary goal is to protect the right and safety and welfare of the subjects and assure that their informed consent process is gone through the proper process. And they should be very much familiar with the protocol. And sometimes I have encountered the investigator is not even aware of exactly what needs to be done as per the protocol. So that the reason why is because he's not familiar with it. And then we come back and when we identify that during the monitoring visit or during the QA audit, then what is the measure we take is go back and train them on protocol. So it's an ongoing, continuous ongoing. Just because we have audited, we have qualified a site does not mean that site is completely responsible. So it's like the responsibility is on the investigator, but the sponsor has the final responsibility where the oversight is the sponsor's responsibility. And FDA keeps sponsor accountable for any issues that comes up in the trial. This is just to say that no investigator can participate in any investigation until they provide the statement, signed statement, which is a Form 1572 in US. This slide talks about the inspection findings from the 2019 matrix. I didn't put all the other findings. So when FDA does an inspection from the BIMO, it's a bio monitoring initiative, and there are requirements which needs to be followed, which needs to be met from the clinical trials, from the sponsors, from the IRBs, and everybody involved in the clinical trials. So as you can see, the most observations, these are all the only ones these are the only major and critical ones which I have included here. And so the way FDA gives is, if there is an official action is indicated, that means it's almost like a warning letter. And they do observe some findings, they do record some findings, but they say that there is no action indicated. That does not mean that the sponsor should not address the finding, but they are not expecting us to respond. The reason why I say that, when they come back to the same site and see the same issue, then that will be a bigger problem. So, and then they will suggest.
Interrupt you?
Pardon
Time is running out.
Yeah. I think I have one more slide, Bhavan.
Okay.
Okay,
thank you.
So, okay, I can rush through. So it's basically the clinical investigator has more number of findings, followed by the IRB and the sponsor. And the SI is the sponsor investigator. So this one lists basically what the findings are, the major findings or the categories of the findings. I don't have to go through that, but again, failure to comply with the form FDA 1572 and protocol deviations and inadequate subject protections. So that's the major thing which comes into picture. And FDA also does the drug quality and oversight trend report. And again, in 2019, there were like 1,258 inspections were done. And they also reviewed the ones which are done by EU authorities from the Mutual Recognition Agreement. And the categories cited are records and reports, laboratory controls, equipment, production, and process controls. So in summary, I would say that we discussed the path from initial candidate identification through preclinical testing, completing clinical trials in humans, and post-trial regulatory approvals by FDA, and the challenges encountered during the process, regulations and guidance that directs through the whole process, and the interactions with the inspections with the regulatory agencies at each stage. And how important it is to interact with the regulatory agencies at each stage of development. And I concluded with inspection findings from both BIMO and the others. Thank you all for your attention, and a special thanks to Professor A.K.D. Bhavani for inviting me to the speaker as a speaker, and Professor Veera Somaya, head of the department. And I'm honored to be here. And for this, for the whole group who has organized this and who made it possible, and the audience who had gone through the painful process of drug approval and drug development and approval process. Thank you all.
Thank you,
and
Dr. Padmaja madam.
questions.
So it is an excellent, very elaborative, informative lecture on clinical trial process for the drug approval. Now, Dr. Padmavati, please moderate the questionnaire session with a 1 or 2 questions.
Okay, madam. Just shall I share the screen, please?
Dr. Padmaja, please close your,
Yeah,
stop your screen sharing.
I did.
Thank you very much, madam, for taking us through this or walking us through clinical trials for drug approval. We have few questions here, but maybe madam asked us to go only for 2 questions. The first one is from Ravinder Dharawat. He says, good evening, madam. Drug discovery to drug marketing takes long period. Then how can COVID-19 vaccination trials are so fast? As these trials, are these trials reliable?
That's a great question. So, so as I mentioned, like, in general, it takes like 10 months for the FDA review and approval. And I also mentioned if it goes through PDUFA or the priority review, it takes 6 months. So since COVID-19 is a pandemic, we all, or like, basically drop everything and do that work. So that's the reason why many companies, many academicians are working on COVID-19 vaccination. So as far as all these, are these trials reliable? I can assure you, and this is something FDA director had mentioned some time back, no matter what, FDA's goal or intention is to ensure the protection. So they're not going to approve something which does not support the data. So that's the reason why they had asked, like, oh, okay, we have done in 3 patients. It seems to be working. No, it seems to be working is not the approach FDA would take, or for that matter, any other regulatory agency would take. It has to provide the support, and it has to provide the, okay, let me put it in this way. Say, for example, if you're doing like in 50 sites, so we're going to audit all of them. If we need 2 quality people to do that, now there will be 20 quality people assigned to that. But we're still going to get that done in the same way as we are supposed to be doing for all other trials. So with the long answer short, yes, these trials, if they're not reliable, the product will not be approved. I can assure you that much.
Thank you, madam. There is one more question from Kirtana, our MSc student. How the clinical trials are carried on during drug repurposing?
That's a loaded question, I would say, but an interesting one. So it depends. It depends on how much repurposing has happened. Say, for example, like if you have changed a small ingredient in the formulation, then again, we have to have that conversation with the FDA. What are thought processes? And also, we have to understand that we are the masters of the product. We know what the product does and what the change of this specific ingredient does to our product. So we should be able to explain that, to convince them, and make suggestions on what kind of trials do we want to do. Is it a big overall 1,000 people trial, or is it like 100 people trial? So, but any suggestion we make or any plan we present, we have to have a supporting data and based on the science. So once science supports the proposal, FDA will agree, but we have to align with them and then conduct it. So I know I didn't give a straight answer, but again, it depends on this particular scenario or the situation we are in.
Thank you, madam. Bhavani, madam, if you allow me, there is one more question left.
Okay, you ask that question.
Yeah, what is the time period to maintain for drug trials? in phase 1, 2, and 3
In general.
in general?
Okay. So generally the phase 1, as I said, within the phase 1, you can make it into phase 1A and 1B also. But the phase 1 is few months, like 3 to 4 months, we can conduct our phase 1 trials. But again, it's hard to say. The reason why I say that is it all depends on how much we know about the product. What are all the information we are trying to gather? If we have already done phase 0 exploratory study and we already know the bioequivalence, then the phase 1 trial will be a shorter period of time. And for the phase 2, it takes quite a bit of time. Maybe I would say it would take at least a few months to a year or two. And for the phase 3, it's many more people, and also it is geographically distributed. So there are local regulations that we need to follow, and we have to abide to those rules and also follow the FDA requirements. So those are the things depends on. So it will take at least a few years, 2 to 4 years. Again, based on the complexity of the product and the target and the disease. If it's a simple disease, then it will take less number of years. But if it is a complex one, then we have to ensure that the safety is determined for the product to send it to the. Have I answered your question?
Thank you, madam.
Hello, Bhavani madam, I have one question.
Okay, you can ask in 1 minute.
Can I ask?
Yes, you can ask, sir.
Dr. Padmaja, this is Dr. Ashok, your classmate.
I know.
Hello.
I know.
Only 1 minute, sir. Because of time constraint.
Hello.
Okay.
Hello. Yes, Ashok, I can hear you.
In clinical trials, success rate is about 11%.
Mhm.
Can rejected candidates become useful after some modification?
Sure, definitely. So when FDA sends that rejection letter, like at the end of after the NDA submission and rejection, they will say what are the deficiencies. If we can prove that the safety and efficacy is maintained,
Yes.
and say, for example, again, if we have a product with a substitution, a fluorine substituent is there, but we modify it and show that that will change the course of action, definitely it is acceptable.
Okay. When we can expect a vaccine for COVID-19?
Many people are asking that question.
Yes.
Yeah. So for Dr. Fauci, who is the head of the NIH and infectious disease department, he's thinking. I mean, the statements made was early 2021.
Oh. Oh, still a lot of time is there.
Honestly speaking, it's very short time. As somebody already asked, how is it reliable? Because there are several things. You can only do so much increasing the number of people, putting a lot of money in there. There is a course of action that needs to happen. Like our reaction, right? You have to do it for 2 hours. It has to be done for 2 hours. Just because we had money and people doesn't mean that the reaction will happen in half hour.
Yeah. Yes.
So that's the reason. So as far as the review process is concerned, that can be expedited. But as far as the trial is concerned, there is only so much we can expedite. But people are making, there is so much involved, so much is going on, and there are multiple people who are involved in that, and they're trying to get this as early as possible.
Okay. Thank you, Dr. Padmaja. Your lecture is very useful and highly informative. and we could know many details about clinical trials for the first time. Thank you so much.
Thank you, Dr. Ashok. Thanks for your
Padmaja,
comment. Appreciate
many
that.
many participants are expressing their doubts. If you permit, we shall share your email, if you permit, with the patients, participants for further queries.
Sure. Anytime. I would be happy to respond to them.
Okay.
Thank you for asking, Bhavani.
Okay. We will provide your email ID to the participants.
Okay.
Okay.
Not a problem.
Thank you. Dr. Ananta, madam, please release the appreciation certificate of Dr. Padmaja Juwadi as a token of gratitude to the Dr. Padmaja. So, Department of Chemistry, Usmania University, Hyderabad, certificate of appreciation presented to the Dr. Padmaja Juwadi for sharing her knowledge and expertise on overview of new drug approvals, a quality perspective in the international webinar on recent advances in organic synthesis and clinical trial process for drug approval.
Thank you.
Token of gratitude to you, Padmaja.
Thank you, Dr. Bhavani, Professor Bhavani. And thank you.
I now request
Dr.
K. Shivkumar to propose a formal vote of thanks to the madam.
Hello.
Dr. Shivkumar?
Yeah, madam. Good evening. Good evening to you all. On behalf of the department, thank you, Dr. J. Padmaja, for an enlightening and knowledge imparting presentation on the overview of the new drug approvals. We appreciate having this mysterious area clarified. Her insightful talk on clinical trials was full of wisdom and information. She explained very well on drug development and approval process and how FDA approval and review process and challenges and risk management, how it's and inspections, how it's going will be inspections will explained very well. And I feel the students and teachers who attended this lecture will be able to gain applicable knowledge. They can follow the tips given in their teaching classrooms and careers. Once again, I thank you, madam, for having spared your precious time for all of us. Thank you, head of the department, BOS, and organizers for giving me opportunity to present vote of thanks for this presentation. Wishing you all the best to be safe from COVID. Thank you, madam.
Thank you, Shivkumar. Thank you, Padmaja, for being with us, for sharing a valuable information regarding clinical trial process and in spite of your busy schedule. Thank you, Padmaja.
Thank you very much, the organizers, and as I mentioned, the Professor Veera Somaya Garu, head of the department, and Professor AKD Bhavani, special thanks. But I would like to extend my thanks to all the organizers of Usmania University who had made this webinar very successful. And I'm glad that I was able to share some of my experiences with you all. And I would be happy to respond to any questions you may have later on via email. Thank you all.
Thank you, Padma. Now we all are enlightened by the lectures given by three eminent speakers. Now I request Dr. P. Sarita Reddy to moderate the feedback session.
Thank you, Bhavani ma'am. I'm sure you all will agree with me that all the sessions were extremely informative and interesting. But we want to hear directly from you as it's time for feedback. So may I request an interesting participants to give their feedback.
Only in 1 minute.
Good evening to all the honorable professors and eminent speakers.
First, you tell your name and organization.
This is Kirtana from Organic Chemistry, MSc Final Year. Osmania University. Amidst of this ongoing pandemic, Department of Chemistry, Osmania University has still providing the opportunity in enhancing and improving our knowledge by organizing the webinars by eminent speakers. As a student, this really helps us to improve our learning in a more interactive and topic-specific way. Today's international webinar on recent advances in organic synthesis and clinical trial process for drug approval was very informative. As in the field of organic chemistry, the synthesis is always challenging and also provides lot of opportunities. As said by one of the scientists, it is important to get results from experiment, but most important is the process of getting that results. The first two lectures has really apprised our minds regarding the scenario of synthesis in organic chemistry. And the third lecture was another wonderful talk on drug development and approval process. Clinical trials are a core component of medical advances, and there are no such things as incurable. There are only things for which man has not found a cure, because of which we are suffering as of now. All the lectures were really very knowledgeable. Before I conclude, I thank all the professors from Department of Chemistry, Osmania University, for their efforts in organizing such wonderful webinars in enlightening the minds of students. Thank you.
Okay. Thank you, Kirtana.
Professor
I think
Bhavani.
even Ravin would like to give his feedback.
Hello.
Goswami.
Hello, this is Dr. Goswami from Gujarat. Ma'am, it is a very useful and a very interactive session, and especially Padmaja ma'am, who explained the complex phase level in a lucid way. So, thank you, ma'am, and thank you also to part 2 lecture who delivered by IIT Roorkee and Mr. Ramaswamy sir.
Ramakrishna.
Ramakrishna sir, and another is from Hyderabad University sir. So thank you very much, all organizer and Osmania University. Thank you very much.
Thank you, Dr. Goswami.
Thank you.
Professor Bhavani. Professor Bhavani.
Yes.
Mr. Pratap Reddy.
Pratap Reddy.
Can I put a word to Padmaja, please?
Yes. Padmaja. It's
Yeah, she's here.
feedback session.
Here.
You can say hi. Hello?
Pratap.
Hello? It's concluding session. So any
Reddy.
participants, Pratap Reddy, just half minute you can talk to Padmaja madam.
She's there. Hi, good.
Here, Pratap.
Good morning.
Good morning. Good morning. Good evening.
It's a typical your style presentation. So nice to listen to you. Very detailed one, informative.
Thank you, Pratap. Thank you for your kind words.
But you disappointed many by saying that the vaccine will come next year.
But we are expecting in October.
I don't believe in false promises, Pratap. I mean, the best thing it would happen is if it comes by the end of the year, but most likely it will be the early next year.
Yeah, it was a nice presentation. Thank you. Please keep it up.
Thank you.
Thank you, Pratap Reddy, sir.
Thank you. Thank you.
Hi, Padmaja. Any participants? Just
Good evening, all the dignitaries.
hello. Who is
Yes, Ravinder, proceed.
okay.
I am Ravinder, a research scholar under Professor D. Ashok sir. A very good evening to eminent speakers, Head Department of Chemistry, BOS, and convener of this international webinar, and all the organizing members, faculty members, and participants. I'm immensely happy and overjoyful to attend this conference and this webinar, because as a research scholar of organic chemistry, I was fully motivated with all these 3 lectures, especially with Professor Ramachari sir, whose contribution towards green synthetic asymmetric methods and catalysis, stereoselective synthesis of new CC bond formation and CX bond formation, and very convenient method, and newly generated catalysts are very reliable as well as very much fruitful, giving fruitful yields. And sir has emerged, uh, done the extraordinary work in the field of catalytic chemistry, and it gives a fully motivated research scholar like us. And the second session by Professor Ramakrishna sir, which is also regarding the same area, that is catalytic chemistry and Diels-Alder reaction. Sir has explained in a very detailed manner with a mechanism, and he gives a simple preparation methods. Even we could not imagine that a research scholar can do the reaction with simple mixing and grinding. Sir has proved that a research scholar can operate the mechanism without using steroids and column chromatography and those things. It is highly motivated research scholars. And finally, Dr. Padmaja Madam gives the detailed clinical trials process. As we don't know, as a scholars, we were studied in our master's levels as a course of as a coursework about clinical trials. But today I would clarify many doubts regarding these clinical trials. And finally, I can say only one word, that this international webinar is a powerful combo of all the clinical trials from synthesis to clinical trial. I would like to thank the Department of Chemistry, organizers of this international webinar, and especially I would like to thank to Bhavani Madam for giving me this opportunity to share my views. Thank you very much.
Okay, thank you, Ravinder. I think we are done with the feedback session. Over to the convener.
Thank you, Satta, for moderating the feedback session. Now we will proceed to the concluding session. Now I request the director of the webinar, Professor P. Veerasubramanian, Head Department of Chemistry, to give the concluding response. Veerasubramanian, sir.
Thank you, Bhavani Madam, for making us to sit 4 hours to hear the lectures, 3 lectures. And I am very much thankful to Professor D.B. Ramachari, Professor Ramakrishna Peddinti, and our colleague, former colleague, Dr. J. Padmaja. Padmaja Madam, many of your classmates, lab mates, colleagues of the department have attended your lecture. Your classmates, particularly Dr. P. Pratap Reddy, Dr. G. Sridevi, Professor B. Ashok, and your colleagues in Women's College, Koti, Professor C.H. Sarla Devi, and your junior in the lab, and my classmate, Dr. V. Ramakrishnan Reddy, also attended your lecture. We are once again expressing our sincere thanks for accepting our invitation and delivering the lecture in the webinar. On behalf of the department and on my own behalf as head Department of Chemistry, we also thank Dr. Satyanarayana, better half of Dr. J. Padmaja, our friend, who always encourages his classmates and friends for academic activities. Thank you, Bhavani Madam, for giving me this chance. Thank you very much.
Thank you, sir, for your response and encouragement throughout this webinar program. Now I request, because time is running out, now I request P. Jalapati, a co-convener, today's webinar, to propose vote of thanks.
This is Dr. Jalapati. Good evening, Head Department of Chemistry, guest of honors, respected invited speakers, teachers of chemistry, scientists, and research scholars, and my dear students. Good evening all. First of all, I would like to thank to the Head of Department, convener of this webinar, for giving me this opportunity to speak on this auspicious occasion, and it is a matter of honor for me. On behalf of Chemistry Department, University College of Science, Osmania University, I would propose hearty vote of thanks. I thank our respected Head Department of Chemistry, Professor P. Veeraswamy Garu, for his constant support and lot of encouragement and also guidance. Thanking you, sir. Also, I would like to extend a special thanks to Board of Studies Chairman, Chemistry, Professor K. Bhaskar sir, and Board of Studies Chairman, Forensic Science, Senior Faculty of Organic Chemistry Specialization, Professor P. Leelavati Garu, for their valuable guidance and cooperation. We have been fortunate to be assisted by our retired senior professor, BSR faculty, Professor D. Ashok Garu. You have been a constant support. You have given us a lot of encouragement and guidance. Thank you, sir. Thank you very much. I also wish to express my gratitude to our Professor B. Manohar Garu, Senior Faculty of Chemistry, and most dynamic President of AUTA for his support and cooperation. I would like to thank our distinguished speakers, eminent speakers, Dr. J. Padmaja Garu, Director, Quality, Maryland, United States of America. Dr. B. Ramachari Garu, Professor of Chemistry, University of Hyderabad. Dr. P. Ramakrishna Garu, IIT Roorkee, for accepting our invitation and making effective, excellent presentations and also making this webinar interesting and meaningful. A special thanks to organizing committee, teaching staff for their unflinching support and coordination. I appreciate the hard work of Professor A.K. Durga Bhavani Garu, who is convener of this webinar. My sincere thanks to Madam. My sincere thanks to all chemistry teachers, working and retired, and scientists, and also postdoctoral fellows, those who are participated in this seminar, in this webinar. Our heartfelt thanks to research scholars, students, and all the participants for active participation and also for making this online conference success. Thank you, one and all. Now, National Anthem. Please display.
अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंधु गुजरात मराठा द्राविड़ विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
Thank you.
Thank you, madam. I am closing the session now.
Durga, congratulations and thank you.
Veeraswamy sir, hearty
No more
congratulations.
chat after this. Sir, no more chat after the anthem ceremonies. I am closing the session.
Close the session.
Thank you. Okay, thank you.